welcome to biology, the scientific study of life. Much of an organism's evolutionary history is documented in its genome. We have seen throughout this chapter that molecular systematics, comparing nucleic acids or other molecules to infer relatedness, is a valuable tool for tracing organisms' evolutionary history. The molecular approach helps us to understand phylogenetic relationships that cannot be measured by non-molecular methods such as comparative anatomy. For example, molecular systematics helps us to uncover evolutionary relationships between groups that have little common ground. For morphological comparisons such as mammals and bacteria, it is possible to reconstruct phylogenes among groups of present-day bacteria and other microorganisms for which we have no fossil record at all. And molecular systematics enables scientists to compare genetic divergence within a species. Molecular biology has helped to extend systematics to evolutionary relationships for above and below the species level, ranging from the major branches of the tree of life to its finest twigs. Still, its findings are often inconclusive, as in cases where taxa divergence at nearly the same time in the distant past. The differences may be apparent, but not the order of their appearance. The ability of molecular trees to encompass both short and long periods of time is based on the fact that different genes evolve at different rates, even in the same evolutionary ranges. Notice that. Boom. For example, the DNA codes for ribosomal RNA or RNA changes relatively slowly, so comparisons of DNA sequences in these genes are useful for investigating relationships between taxa that diverged hundreds of millions of years ago. Studies of rRNA sequences, for example, indicate that fungi are more closely related to animals than to green plants. Notice this figure. Boom. In contrast, the DNA in mitochondria, mtDNA, evolves relatively rapidly and can be used to explore recent evolutionary events. One research team has traced the relationships among Native American groups through their mtDNA sequences. The molecular findings corroborate other evidence that the Paima of Arizona, the Maya of Mexico, and the Yanomami of Venezuela are closely related, probably descending from the first of three waves of immigrants that crossed the Bering land bridge from Asia to the Americas about 13,000 years ago. Gene duplications and gene families. Gene duplication is one of the most important types of mutations in evolution because it increases the number of genes in the genome, providing opportunities for further evolutionary changes. The molecular phylogenes for gene duplications and the influence of these duplications on genome evolution can now be followed in detail. These phylogenes must account for repeated duplications that have resulted in gene families, which are groups of related genes within an organism's genome. Notice this figure, et voila. Like homologous genes in different species, these duplicated genes have a common ancestor. We distinguish these types of homologous genes by different names, orthologous genes and paralogous genes. The term orthologous genes, from the Greek orthos, straight, refers to homologous genes that are passed in a straight line from one generation to the next, but have ended up in different gene pools because of speciation. Notice that. Speciation, orthologous, and gene duplication. Notice that. Paralogous gene. In chapter 23, we encountered an example the olfactory receptor genes, which have undergone many gene duplications in vertebrate animals. Humans and mice each have huge families to more than 1,000 of these paralogous genes. It is possible to describe most of genes that make up genomes as representing one of these two types of homology. Note that orthologous genes can only diverge after speciation has taken place, with the result that the genes are found in separate gene pools. For example, humans and mice each have one functioning P homoglobin gene. These genes serve similar functions, but their sequences have diverged since the time that humans and mice had a common ancestor. Paralogous genes, on the other hand, can diverge even while they are in the same gene pool, because they are present in more than one copy in the genome. The paralogous genes comprising the olfactory receptor gene family in humans have diverged from each other during our long evolutionary history. They now specify proteins that confer sensitivity to a wide variety of odors, ranging from various foods to sex pheromones. Genome evolution. Now that we can compare the entire genomes of different organisms, including our own, two remarkable facts have emerged. First, orthologous genes are widespread and can extend over huge evolutionary distances. 99% of the genes of humans and mice are detectably orthologous, and 50% of our genes are orthologous with those of yeast. This remarkable commonality demonstrates that all living organisms share many biochemical and developmental pathways. And second, the number of genes seems not to have increased through duplication at the same rate as phenotypic complexity. Humans have only about 5 times as many genes as yeast, a simple single-celled eukaryote. Even though, unlike yeast cells, we have a large complex brain and a body that contains more than 200 different types of tissues. Evidence is emerging that many human genes are more versatile than those of yeast and are able to carry out a wide variety of tasks in various body tissues. Before us lies a huge and existing scientific challenge, unraveling the mechanisms that enable this genomic versatility.